All right, so welcome to this week's Ronin seminar. We got Gordon Webster, who is going to tell us why, or perhaps whether there really is more to life science data than being really ridiculously good looking. Please, Gordon, take it away.
Thank you. Hi, I'm Gordon. I've been a Ronin for a couple of years now, and I work in the field of computational and structural biology. And most of the work that I do is through my company. We have with a fellow Ronin, Alex Lancaster. We have a small digital biology consulting firm, and we do a lot of work in computational modeling. And that's sort of what I'm going to talk about today in this talk, and sort of why that's difficult in biology. So I'm very interested in the role of models in science, particularly in my own field of biology, but also, you know, models more generally in sort of rational thought. So when I use the term model here in the talk, I'm mainly going to be talking about computational models, but sometimes I'll be using model in the broader sense of a mental or intellectual construct that we use to, you know, organize data and rationalize our experience of the world. And in a way, I mean, all scientific theories are kind of implicit models that we use to explain, you know, our observations of the world. But I'm mainly going to be talking about our implicit models. I'm going to be mainly talking about explicit models. So you know, actual models using computers and data from scientific experiments, models of causal processes that can explain data and that we can use to generate and test new hypotheses. I'm going to try not to make it too sort of biological, but if anyone who isn't a biologist has questions about any of the biology, or if I skip over something that you feel is important, then, you know, feel free to interrupt me, and I'm happy to explain it. So as I said, my business partner and fellow Ronin, Alex Lancaster, and I are actually doing the kind of modeling work that I'm going to describe here in commercial applications for real paying clients. So we are both proof that it's still possible to pursue a career in modeling even when you're a middle-aged guy with an expanding waistline and graying hair. So the theme of this talk is really about the challenges of modeling biological systems. And it's very difficult. The causal and mechanistic modeling of biological systems is very difficult, mainly because of the complexity of the systems that biologists have to deal with. And so a lot of people will you hear a lot about big data, and big data has worked really well in other fields. I mean, if you're in advertising and things like that, you're sort of running online ads and doing sort of analytics of, you know, do people click on a Tuesday night more than on a Wednesday night, and, you know, what kind of visuals in the ad, you know, give you a better response, that sort of thing. I mean, big data works pretty well there, and a lot of people in biology have been sort of rushing to embrace big data, and it hasn't really panned out, and I'll talk more about that in a minute. So what you'll often see in a lot of big pharma companies, a lot of the ones that I've either worked at or consulted at, are these sort of data dashboards like you see here. So a lot of people are doing these kind of pretty data visualizations, and, you know, they're not without value. I'm not kind of bashing them sort of without restraint. I mean, they have a value. If you're looking for trends in your data and you want to make just nice PowerPoint slides of your data, then these are really valuable tools. But in terms of actually deriving any insight from them, they have not been very successful at all. And in a way, these data visualizations, you know, they kind of really are kind of an effort to sidestep the complexity of considering causality in these complex biological systems, which I'll talk more about in a minute. So these data visualizations are pretty, but you really need models to make sense of your data. And this is a scene from the famous movie Zoolander, where two male models have to find some secret files, and they're told they're in the computer, and they start trying to figure out how to physically crack open the computer. And the scene, I don't know if you've all seen it, the scene ends in a wonderful homage to 2001, where all the monkeys are like fighting and screaming and hitting each other with bones and stuff. But I promise to keep the Zoolander references to a minimum from here on in. So a couple of years ago, I published an article, the one on the left, Big Data Does Not Equal Big Knowledge. And it was really about the hype in big data in the life sciences. And I was surprised that I didn't get a lot of pushback on it. I thought that I would. It actually says in the opening sentence that the risk of alienating half my audience here, I'm going to open with the statement that the life science big data scene is largely big hype. And actually this year, so in the meantime, IBM have been pursuing their kind of big data approach, which is primarily built around AI and their Watson platform. And they were very excited about this and talked about this being even, perhaps the core of their business platform in the future. But again, in biology, it really hasn't panned out at all. And so this year, I actually wrote a kind of follow-up article in which I referred to my previous article of a couple of years ago. And this was triggered by the fact that IBM basically slashed its life science program, big data with AI kind of initiative in Watson, and laid off a lot of its staff. I don't think the program's entirely dead, but essentially it's been a miserable failure in biology. It's Watson can still beat a human being at Jeopardy, but it can't tell oncologists anything that they don't know already about how to treat patients with cancer. So this kind of, again, is sort of a big data approach. And what I'm characterizing here as a big data approaches are approaches where you're basically either statistically or semantically sifting a lot of data to look for trends in the data that can, reveal how the underlying system that produced the data works, but without really attempting a sort of a causal or mechanistic explanation. So in other words, you're kind of trying to avoid getting into causality or mechanistic hypotheses altogether. And I think that, given the complexity of biological systems, there was a hope that, if you had enough data and it was carefully enough measured and you measured it under a variety of different circumstances, that the trends that the data would allow you to see would somehow, give you a kind of a handle on the way that those underlying systems behaved. And I mean, at least to date, it really hasn't panned out. And it certainly hasn't panned out with AI. And with the case of IBM, you can be sure that it wasn't for a lack of funding or a lack of really brilliant minds in AI, trying to make this work. So I'm a firm believer. So in both of those articles, I come back to the fact that I think that models are the way to go, and that causal and mechanistic models in particular are very powerful. But at the same time, it's very understandable why they haven't really caught on in biology. And I'll talk more about that in a minute. But the first thing to say about models is that good models explain data. So when Watson and Crick first came up with the structural model for DNA, and they looked at the complementary strands on the double helix, they immediately realized, well, here's the mechanism for heredity. And so, the model itself basically explained not just the data, but a whole world of phenomena that we see in biology. And it's kind of an implicit model that we all use today, whether you're doing like PCR or sequencing or whatever. Everybody's working with this underlying model of DNA as this sort of, double-stranded complementary structure. And actually, a lot of people don't realize that the DNA structure, the model that they created, for which they won the Nobel Prize in 1962, was actually still just a model until the 1970s, when somebody actually solved a crystal structure of DNA and actually, saw the double helix. Until then, it was kind of derived from a low resolution a set of fiber diffraction experiments, but nobody had actually really ever truly seen the double helix until the 1970s. So it was actually a model, but it was such a good model, and it explained so much about biology that everybody felt it just had to be right, and indeed it was. But it's also true that bad models can explain data too. So sort of, if I can take a little detour into astronomy. So before Copernicus and Galileo, a lot of Western cosmology was kind of determined by scripture and the church, and they favored the sort of geocentric cosmology of Ptolemy, where the Earth was at the center of the cosmos and everything orbited around it. And in this model, one of the phenomena that we see, that we take for granted now in our heliocentric model of the cosmos, sometimes the Earth, as it orbits the sun, we overtake Mars on the inside track, as it were. And so Mars appears to sort of go backwards in the sky at those points. And in our heliocentric model, this is not a problem. But of course, in the geocentric model, they had to come up with some way to explain the retrograde motion of Mars. And one of the ways that they did this was to have Mars orbiting in these things they called epicycles, where it orbits around the Earth, but also kind of does these little sort of loop-the-loops going around its orbit as well, so that when it's moving backwards in its smaller loop, this is the explanation that was given for the retrograde motion for Mars. So this was an example of a bad model that was essentially being used to explain the observations. And of course, we now know that this is not a correct model. But it's also true that bad models can advance scientific discovery as well. So again, staying with the astronomy examples, astronomers often combine observations with Newtonian gravitational models. So they'll make a bunch of measurements of a position of, like, this is a binary star system here, and then they'll fit the observations to the Newtonian gravitational model on a computer. And oftentimes you'll see discrepancy between the actual observed orbits of the stars and the Newtonian model, and the discrepancy between the observations and the model can actually reveal missing mass, and this can lead to and has led to the discovery of new planets. So the amplitude and the direction of the discrepancy can even be used to calculate the mass and the position of the orbiting planet that's too distant and faint to be observed with a telescope. So it's something that I think a lot of people who are not working with models a lot don't often realize, is that even models that are wrong or incomplete can be very valuable, because the discrepancy between the model itself and the data are a kind of a knowledge, if you like. And this is particularly relevant in biology, because I think a lot of biologists are put off from using models because there's the feeling of, well, biological models are always going to be incomplete just because of the astronomical complexity of a cell and the number of moving parts it has and all this sort of thing. A model that's either wrong or incomplete can actually be very useful too. And models can do a lot more than just prediction. So when we talk about models, again, people who are not sort of familiar with working with models a lot will tend to think of models as being, something like weather forecasting and prediction. So this kind of predictive forecasting is what most people think of when they think about models. So I guess weather forecasting is probably the place where most people have some sort of daily brush with models. Is it going to be cold? But I mean, models are also used extensively in civil engineering. So there's no bridges or large structures really get built these days without being built in a computer first. And people will often design the components with CAD, and then they'll go and build the components in real life, and they'll test them, and the data will be fed back into the computer model. And so there's a kind of a nice a kind of a cycle between the sort of the virtual design and the real design of the structure. And then, of course, there are countless other examples like acoustics. So people are using computer models now when they design new symphony halls and things like this, sort of looking at how the acoustics of the building will behave before the building is ever actually constructed. So this is one area that you can use models, but they can also be used to generate and test hypotheses. If you think about the astronomy example that I gave earlier, I mean, the discrepancies between the model and the data are seeds for new hypotheses there. So in that case, it was a missing planet. And the example I'm showing now is a cell signaling pathway. And for those of you who are not biologists, this is basically kind of a cartoon of the way that the biologists imagine that cells work internally. These are kind of like the circuits in cells. And so that structure at the top is a receptor on the surface of a cell, and it has a ligand which is bound to it, which is the little kind of circular thing resting in that cup there. I don't know if you can see my mouse arrow at all. And then that triggers some changes on the leg of the receptor. You get these changes, the post-translational modifications of the receptor, which are basically the attachment of usually phosphate groups to the protein leg of the receptor. And then this propagates down through this chain of cause and effect where all these proteins start modifying one another, and you get a signal that can end up, for example, in the expression of a gene. And oftentimes these pathways are very carefully regulated, modulated. Evolution has produced sort of incredible circuits that that can have positive and negative feedback and all sorts of control systems of the kind that we build into the systems that we construct ourselves electronically. And so here, for example, I've put an arrow saying, you know, is there negative feedback in this particular part of the loop? And this is the kind of thing that you can test with a model. And models are also a wonderful vehicle for the communication of ideas. So scientific collaborations generally involve some kind of sharing and comparison of data. And models are great frameworks that you can use to organize the data and discuss them. And it's nice because rather than just kind of looking at the data in an isolated sense, as you know, as often happens in science, the model is a way to sort of integrate the data together and to discuss it in a kind of more holistic manner. And some of the companies that we've worked for, we've actually seen this firsthand. So people have an awful lot of data in companies. Usually the problem in most of the life science companies that we talk to is that they're kind of drowning in data, and a lot of it feels very disparate and isolated and unconnected. And it's quite an epiphany to see all that data put into a model and see it organized in a model and see how it all fits together and how the whole thing kind of works. So models really are an amazing way to take data and organize it and think about it in a more integrative manner. And what I want to talk about today, the examples I'm going to talk about are cell signaling networks. So this is, again, sort of a cartoon like the one I just showed on the blackboard in the earlier slide. So this is the epidermal growth factor receptor pathway. That doesn't really matter. I'm going to try not to make it too biological, but for any of you who are not biologists, this is kind of like the way that a cell works. I would say that this is a sort of a consensus view among biologists. If you ask most biologists how a cell works internally, they'll probably draw you a cartoon like this. I wouldn't want anyone who's not a biologist to think that this implies the existence of circuits of the kind that we would build in computers. These lines between the different components in the pathway are more like interactions than actual circuits. So a better analogy than a circuit is more like the sharing of gossip in a crowded room. So if you imagine a big party with a crowded room full of people, and somebody on one edge of the room comes in the door and shares a piece of gossip with somebody, and then that somebody passes the gossip on to someone else, the interactions that you'll see, the way that the gossip spreads through the room, this is kind of more like the way that cells work. And so the interactions in the room will be more decided by who are friends with other people in the room, and how many people are clustered together, and how many people get to share the gossip at the same time. I mean, it's not a perfect analogy, but it's a better analogy than thinking of hardwired circuits. So I just kind of throw that out there for anybody who's not a biologist, that when we talk about these cell signaling pathways, we're not really talking about networks like a circuit, but more like networks of people in a room talking. And so a lot of the signals that they're passing in the biological pathway are things called post-translational modifications. And again, for those of you who aren't biologists, this basically means attaching little chemical groups to proteins as kind of signals. So mostly these are phosphate groups, and there are proteins called kinases that are abundant in cells. And these kinases will basically attach to a protein and attach a small phosphate group, which is then like a sort of semaphore. And then that protein will go away and interact with other proteins in other parts of the pathway. And whether or not that protein carries that particular flag or semaphore of the little phosphate group will determine how the other proteins in the pathway will interact with it, and so on. And this is basically the kind of network that I'm going to describe. So these are things that are very hard to model. And I'll talk about why that is in a minute in more detail. But one of the problems that we have in biology is that most of the modeling paradigms that we use are borrowed from other fields like sort of physics and chemistry and so on. And calculus has been around for like 300 years. It's still the kind of lingua franca for modeling in biology as well, which is kind of unfortunate because it's not really an ideal paradigm for modeling biological systems, and I'll talk about why in a minute. But it's a very powerful form of a very powerful way to model dynamic systems. It's tried and tested in other areas. I mean, it's been used for planetary orbits and fluid dynamics and the behavior of suspension bridges. I mean, there's almost no area of dynamics to which calculus has not been applied. And so, it's out there. It's a tried and tested and powerful tool, but unfortunately, it really doesn't lend itself to biology. And one of the reasons is if you look at this is part of a model that we actually built for a client recently. We didn't build the mathematical model. We built it a different way. This is a model taken from a publication. And if you look here, if you look at the top, at the diagram at the top, you can see this is a model of a protein called C3b, which has these little white bubbles on it, are sites on the protein. And the purple bubbles are states of those sites. And so this is a model of the rate of production of this protein called C3b in the fluid phase, which is indicated by that little purple F in the purple bubble there. And if you look at the big formula, I'm not going to go through it or anything, but if you look at the very topmost part of it before the equal sign, this basically just describes the rate of change of this particular species in the pathway over time. So the D, the square brackets denote concentration. So this is the rate of change of concentration of FC3B over time. So over DT means just over time. And you look at all these different terms here. Each one of these is a process. So it's a rate. The Ks are for the most part rates at which things happen. And then all the terms in the little square brackets after these are other components of the pathway. So the rate of production of C3b has all these different terms in here. So this is actually a model of a part of the immune system that controls the way that the immune system responds to an infection. So you'll see the word pathogen and things like that in there. And I think one of the first things you notice is that it's very opaque. It doesn't look like biology at all. And even if you're an experienced modeler, experienced using these kind of calculus models, they're called ordinary differential equations for modeling biology, it's not easy at all to read the biology in these models. So they're not very transparent with regards to the biology. They're also very error prone. They're something of a house of cards in the sense that any one of these terms in here, each one of these little terms that you see like in the square brackets will also have a similar equation for it. So you need an equation for every single one of these sort of components that you see in the square brackets in here. So each one of those will also have an equation like this with a whole ton of other components that it depends on. And so it's kind of a house of cards. If you make a mistake in one of these areas, in one of these components, your mistakes propagate through the entire model. So these models are in addition to not being very transparent, they're not very robust either. And there's a more significant reason why they're a very poor paradigm for building biological models, which I'll talk about now. So again, without needing to go into all the math and everything, the take-home message for using these ordinary differential equation models is that you need an equation like the one we just saw for every single species that can possibly exist in your model. And I'm going to give you a really simple example of the simplest sort of part of a pathway, which just consists of a kinase, which is one of those proteins I described that puts these chemical groups on other proteins. The protein that the kinase puts the chemical group on is referred to as a substrate, and these have sites at which these kinases can attach these chemical groups. And so here I've created the simplest signaling system you could possibly think of, which is just a kinase and a substrate, which has two sites, A and B. And already in this simple system, there are 13 possible species that you can form. So you can have the kinase on its own, just floating around unbound. You can have the substrate floating around unbound, but of course the substrate has two sites, each of which can be phosphorylated. So it has 4 possible phosphorylation states. So you could have the substrate completely unphosphorylated, or with A phosphorylated, or with B phosphorylated, or with A and B phosphorylated. And then, of course, all those states can exist with the kinase bound at A in one of those 4 states, or with the kinase B bound at B in one of those 4 states. So this very, very simple model already requires 13 equations, and you've only just, basically modeled the very first 2 components in your cell signaling pathway. And if you add the phosphatase, so phosphatase is like the opposite brother of a kinase. The phosphatase undoes what the kinase does. The kinase adds phosphate groups to a substrate, and the phosphatase removes them. And just adding, we won't go through all the different states, but just adding the phosphatase to this system, the complementary phosphatase, requires you to rewrite all 13 of the rate equations that we would need for the first part of the model, and adding 21 new ones. So already you kind of get the idea. This is called a combinatorial explosion of states. And if you look at the real pathways in cells, so in the diagram on the right, this is the top part. This is only the very uppermost part of the epidermal growth factor receptor signaling pathway, which is a very important pathway. People are very interested in this because it seems to be very central in a lot of cancers. And if you look at this pathway, the very top part of it, just in the complex between 2 of these receptors, EGFR and ERB3, there are 3.8 times 10 to the 9 states just in one complex formed by those 2 receptors. And in 2 EGFR receptors combining together, there are actually 3.8 times 10 to the 10 states. And as a kind of an exercise at a startup I worked at, where we were building models of the epidermal growth factor receptor pathway, we did the math and figured out that there are actually about 10 to the 33 possible species of molecules that can be formed just in that one pathway. And remember, this is only one pathway in a cell that has, hundreds and hundreds of different kind of pathways. So there's no way that you can ever really build an accurate model with ordinary differential equations of this pathway. And so this is a kind of a much more significant reason why, using calculus to build dynamic models of these complex biological systems is really a non-starter. And I coined a term, uncertainty principle for mathematical modeling in biology, which says that you can have scope or resolution, but not both at once. And what I mean by that is what typically happens is people will build models of these pathways using ordinary differential equations, but in order to simplify the models, they'll reduce the number of sites down. So for example, the epidermal growth factor receptor itself has 9 phosphorylation sites on the tail of the receptor, and they'll reduce those down to 1. And so they'll sort of model what essentially should be 9 separate states with 1 state. And so the model is already becoming kind of low resolution. And there's a really a sort of a scientific problem here too, and that is that you have to sort of a priori make a decision about which parts of the model are important, which parts are not for the behavior of the system as a whole, in order to make these simplifying assumptions. So that's already kind of a problem that affects the scientific integrity, if you like, of the modeling that you're trying to do. Another problem with calculus and differential equation models is that they are deterministic models. So it's a mathematical, analytical model that's produced. Deterministic models have outcomes that are precisely determined through known relationships among states and events, without any room for random variation. A given input will always produce the same output. So these are kind of work in a very, they model a very kind of idealized world where everything's kind of smooth and continuous. There's no noise. They work well where the statistics of large numbers are applicable, which is true if you're doing a chemical reaction where you have hundreds of millions of molecules in a well mixed solution. But in a cell where you might only have maybe 10 copies of a gene expression is seldom going to be smooth and give you that kind of smooth analytical kind of curve. It's usually going to be very stochastic and noisy. And that brings us to stochastic models, which possess some inherent randomness and can represent the noise in a system. And the same set of parameter values and initial conditions will lead to an ensemble of different outputs. So a stochastic model is a result that includes system noise. And these models work well whether or not the statistics of large numbers are applicable. So stochastic models, for example, are a much better way to model gene expression than ordinary differential equations. and they capture the discrete heterogeneous stochastic behavior that is typical in biology. So if you take a bunch of identical cells and you kind of freeze them at some particular point in their life cycle so that they're all synchronized, and then you somehow release them either by thawing them or by chemically freeing up some components so that they can start, the pathways in them can start working. And then you go back to that population of cells every sort of periodically, and you kind of sample the cells and have a look at what they're doing. You'll find that they're all in different states, especially with regard to things like gene expression and levels of proteins and levels of activity of certain proteins, phosphorylation, and so on. And this is because biological systems are inherently stochastic, and what you see is a kind of a population of cells that are, you know, very, very often very different from one another. You don't get the kind of idealized, you know, synchronized systems that you'd sort of see if you were to model these cells using sort of mathematical equations. And there's one important issue is that stochastic processes more and more are being recognized as being very important in biology. So stochasticity in gene expression, for example, produces the pattern diversity that you see in butterfly wings. It also affects, for any of you who are not biologists, you might not realize that bacteria themselves can be infected. And there are things called bacteriophages, which are viruses that actually infect bacteria. There's a very well-studied phage called phage lambda that will infect E. coli, and it has two stages in its life cycle. In the default stage, it will infect the bacterium, and it will kind of integrate its genome into the bacteria and just kind of go along for the ride. It's called the lysogenic phase. So basically what will happen is that the bacteria will divide, and unwittingly, as it divides and reproduces its genome, it will reproduce the phage genome with it. And so the genome is just the phage is kind of like a stealth parasite just along for the ride. But then if things get bad for the bacteria, like it's exposed to ultraviolet or some kind of like chemical irritant that threatens the integrity of the bacteria, there are certain responses that the bacteria will switch on, which then alert the phage proteins to the fact that something's happening, and the phage there's a switch that will flip over in lambda, and it will switch to what's called the lytic form, in which it will frantically start making copies of itself and packaging them up in viral protein. And basically it will lyse the cell, which means just kind of splitting it open, and millions of lambda phage will be released to go away and infect other bacteria. So kind of like rats leaving a sinking ship. And the pathway in this in phage lambda is very interesting because it's inherently unstable and designed to be so. So if you look at populations of bacteria infected with phage lambda, you'll see some of them will be in the lysogenic stage and some of them will be in the lytic stage. This is kind of a hedging strategy where the phage is basically able to in a moment's notice to flip from one form to another, and even to have some diversity in the population of phage, so that it's not making all its bets on, for example, the lysogenic phase or the lytic phase. And this also happens in yeast. You can see that that yeast have non-genetic diversity that's conferred to them that allows them to develop antibiotic resistance, again from stochastic gene expression. So stochastic processes and noise are very important in biology, and there are countless other examples. And I think this is an area that's being more and more studied now. People are starting to realize how important noise is. So there's a tendency to think of noise in terms of the kind of noise like that interrupts your cell phone call or your music when you're streaming or something like that. But noise can actually be very useful. I just want to talk about one other thing that I find very interesting in modeling, and that is the sort of idea of cognitive impedance. So most biologists are not mathematicians, and so most of us, even who have done some math and have built models using calculus, don't really enjoy this kind of modeling, because it's not the way. If you ask a biologist how a cell works, they'll probably give you a cartoon like the one shown in the top left there. And if you ask a mathematician about that system, they'll give you a system of equations. And if you look at the output from the system, the way a biologist would typically look at the behavior of that system in the lab would be to do something like a Western blot. So on the top right is this would be like the sort of the result of an experiment that the biologist does in the lab, looking at a behavior of the system on the left. So these are proteins which are put onto a gel and kind of run in an electric current which separates the proteins out. And you can measure the relative abundance of the proteins from the gel by how sort of dark the bands are that appear on the gel. And again, if you look at the mathematical output, you'll see something more like a kind of a graph shown in the bottom right. And these are all valid. It's not a question of none of these kind of idioms being valid or invalid. But if you're a biologist, the top row is kind of how you're used to thinking about the systems you're working with and how you tend to view the data from them. And the bottom row is more like the way a mathematician would view it. And so one of the things that's important in modeling is to be able to use the model to sort of develop a cognitive connection with the system that you're studying, because that's where insight really comes from. And so models that don't allow the scientist to develop a cognitive connection with the data tend not to be as useful for developing insight. So this is the approach that we like to use in our work. This is I'm not going to go through all of this stuff, but this is a programming language for biological modeling called kappa. And this allows us to build stochastic models without using mathematics. It does have a kind of semantic formalism to it, which is also, frankly, a little computer sciencey. The way I like to think about this is if you think about PostScript. So PostScript is actually the underlying sort of architecture of most printers these days, and nobody creates a document in PostScript. We use a word processor like Microsoft Word, and it's WYSIWYG, so what you see is what you get. You can see what the whole thing looks like as you're typing on the screen. You can see the fonts and the layouts and all that kind of thing. And then eventually, under the hood, all the magic is done on the printer with PostScript, which is a very powerful semantic formalism that basically allows printers to print on pages of any size and to scale things up and down, because everything's sort of like vector graphics and there's all sorts of great stuff happens under the hood. But the idea of actually writing a document in PostScript is kind of not very appealing. And so we have word processors that do that for us. And I like to think of kappa as potentially becoming like the PostScript of biology at some point, where we would have productivity applications built over the top of this that would be more biological, if you like. And then under the hood, we have this semantic formalism. But basically, kappa allows us to build models of these pathways using only 5 very simple kinds of rules, which are synthesis, degradation, binding, unbinding, and modification. And again, I'm not going to go through all the syntax, but if you look at the kappa rules, they're actually composed of agents. So this is depending on whether you decide that the agents are the most important part of the model or the rules are the most important part of the model. People refer to this as agent-based modeling or rule-based modeling. And you can see that the agents in the model are this is the kinase and the substrate again that we saw earlier. So K and S are the 2 agents, and these agents have sites through which the agents are able to interact, for example, by binding, as in the example on the third one down shows a binding reaction. And the sites are also able to have states that can be modified. And it turns out that with these 5 simple kinds of rules, you can model pretty much any kind of cell signaling or metabolic pathway. Yeah, I'm not going to go through all the formalism of it, but that's just an example of how we construct models. And this allows us to do some really neat things that are also not very easy to do with mathematical models. So we can do a dynamic analysis. So the graph in the middle shows the result of a simulation. So one of the things we can do is we can assemble a simulation of the pathway that we want to model, which is kind of like assembling all the players of different kinds, putting them on the field. You have a set of starting conditions, which determines how many kinds of each player you have and what state they're all in. And then essentially you blow the whistle and you see what happens. And so that kind of simulation, which I'll talk about in a minute, is shown in this middle graph here. So this is what you get. So that's an example of a dynamic analysis that you can do with this kind of modeling approach. but we can also do really nice static analysis, which is the kind of analysis that if any of you are familiar with computer compilers, if you write a piece of code that has an error, the compiler will often tell you, oh, this line here has an error, or if you run the program and you get an error that occurs at a certain point, it will tell you where in the code it occurred, and it will tell you which routine was calling that particular line of code. So on the right in the slide you're looking at now, we can now do that biologically using kappa. So you can say, for example, I want to see whenever a certain gene transcription factor complex gets formed, whenever that event occurs, show me how we got there. And then what the simulation will do is it will store the pathway by which that gene transcription complex was formed. And as soon as that event occurs, it will kind of give you what a computer scientist would call a traceback. So these are some of the neat things that we can do with the systems that we're studying when we're using this kind of modeling. And this is a result of this powerful semantic formalism. So in a way, it's a kind of a compromise that you have a very powerful system that allows you to do this kind of work, but the syntax may not be very biological. So this is kind of one of the compromises we often have to make in modeling, which is why it would be nice not to have to build models directly in kappa. So I was actually part of a startup where we were working on this project until 2010, when we had the rug pulled out from under us by the crash. And so these were the kind of things we were building. So on the left here, you can see this is actually a rule and agent generating interface. So this allows you to build those kind of kappa rule. Yeah, these allow you. We were building a kind of a cloud-based collaborative computing platform that allowed you to build these kind of kappa models online and also build them collaboratively. And so we had this really nice interface where you could actually assemble these sort of cartoons, and you could build the rules graphically. And then the outputs that we produced were also, we made them more biological. So if you look at the graph on the right, there's an interesting story to this graph. So the simulation software itself produced the graph you see at the top there, the sort of the dark part with the different lines. And I had a suggestion of adding essentially what would look like the Western blot part, which is the thing that the biologists would see in the lab. So if we go back to this slide here, so you see on the top right there, that's what a Western blot looks like. And that's what a biologist would be used to seeing as an output from their experiment. And so my idea was that we add something like that underneath the graph. So this kind of vertical bar that runs across here, you could drag this across the graph, and if you dragged it across the graph, it would simulate for that particular time point what the Western blot would look like for each one of these observed proteins. And the people who wrote the code initially thought this was kind of, why would you do this? Because the data is already, there's a much higher resolution in the data in the graph than there is in this Western blot. But when we tried it out with biologists, they were extremely excited by it, and it sort of allowed them to make this cognitive connection between the model and their own data and their own experiments, which they found very exciting. And none of the biologists that we tried this with had actually been using modeling themselves. And so I wrote a piece inspired by this experience a while ago called "The Central Role of User Experience Design in Scientific Modeling." And I said that success of a modeling approach would depend in no small part on the appeal that it could make to the imagination and intuition of the biologist. With computational modeling, as with software development, even the most meticulously crafted of tools will have little or no impact or utility in its field if a cognitively dissonant user experience results in it rarely or never being used. So again, this is one of the problems in biology, is that a lot of the tools that people have traditionally used for biological modeling just do not lend themselves very well to the idiom that biologists are used to think in. This is a very busy slide. It's kind of a description of the algorithm that we use for the simulation part. And I think I'm going to skip it unless anybody's really interested in it. If somebody wants me to come back to this, I will. But this is essentially the description of the algorithm that we use to produce the simulation. So we take all the rules and the agents that we develop in kappa, And this is the algorithm that we use under the hood to produce the dynamic simulations. And I can come back to this if anyone's interested afterwards, but in the interest of time, I'm going to move forward because I only got a few minutes left and I want to get to the practical example. So this is what a kappa model looks like. I just wanted to sort of, before I finish, I just wanted to show you what a real model looks like. This is a very simple system of a kinase and a phosphatase competing to either phosphorylate or dephosphorylate a substrate. And this is, as I said, a very common motif in cell signaling pathways. It's abundant in cells. You'll find this motif everywhere. And what this motif does in cell signaling pathways is it allows cells to respond very, very strongly to a very weak stimulus, which, if you understand the biochemistry of the way these cells work, know that that's not an easy thing to achieve using sort of the normal enzymes and things. So this is kind of a cellular switch. It's a bistable switch that has sort of different states depending on the relative levels of kinase and phosphatase substrate in there. So you can see in the model on the left, again, I'm not going to go through all the kappa, but you can see you have a list of the agents. You have K, which is the kinase, P is the phosphatase, and S is the substrate. And each one of these has a site X, and in the substrate, the site can have two states, U, which is unphosphorylated, or P, which is phosphorylated. And these are just labels. They can mean anything that you want them to mean when you're building the model. And then you have the concentrations of each of these. You'll see that the substrate is actually tenfold higher concentration than the kinase variable called fraction K, which we use to control the level of kinase. So when it's 1, the kinase and the phosphatase are both present in the model at equal concentrations. If I adjust it up or down, I can adjust the level of kinase and phosphatase, the relative levels of kinase to phosphatase up or down using that variable. Then there's a section underneath, which is just the conversion from concentrations, molar concentrations, to numbers of molecules, because in these stochastic models, every molecule is an entity. So the model actually consists of a pool of these molecules, and there are actual countable numbers of molecules. This is not a model of a bulk system like an ordinary differential equation model. The next section is the rates. So these are the rates that all these processes happen at. And the rates are going to be applied to the rules which are underneath. And if you look in the section of the rules, we just take the top rule, for example, KS assoc, which is the rule, a reversible rule that says that the kinase can bind the substrate at site X and it unbinds. So you can put either a forward arrow or a double-headed arrow. The double-headed arrow implies that this rule is reversible. And so there are two rates here, an on rate and an off rate. And that's really how simple it is to define a rule using this system. And then in order to start the simulation, all you need are a set of initial conditions, which are down here. You can see the concentration multiplied by a factor that converts the molar concentration to the number of molecules in the simulation. And then at the very end, you have a set of things called observables, which are just the things that you want to track in the system. So in our case, we're interested in the relative levels of unphosphorylated substrate and phosphorylated substrate. If we run the model with fraction K set to 1, this is what we get. So we have equal levels of kinase and phosphatase activity in the system, and the system, all the substrate is initially unphosphorylated. So you can see the blue line starts very high, and the orange line, which is the phosphorylated substrate, starts at 0. And very quickly you establish an equilibrium where there are roughly equal amounts of phosphorylated and unphosphorylated substrate. And just like, again, one of the things with the ordinary differential equation models is you get two perfectly smooth curves if you built this model using ordinary differential equations. And by the time you got out to about sort of 40,000 here, the lines would just be completely flat and nothing would be going on. And as we know from all our thermodynamics textbooks, a state of equilibrium in a system like this doesn't mean that no change is occurring. It just means that there's no net change in the levels. But if you look at this close up in this little window at the top here, you can see that an equilibrium state in a stochastic model shows you kind of what you would see in the cell, and that is that all these processes are still going on, but that the net change is effectively around a mean of 0. You can see that very clearly here in this sort of blow up of the of the curve at the equilibrium condition. So this is again a nice feature of stochastic models, is that you see the real noise of the system. You see how the system behaves exactly as you would if you were to actually take cells and measure this activity in real cells. And now in this next slide, I raised the fraction k. I set the value to 1.1. So that means there's 10% more kinase now than there is phosphatase. And you can see there's this huge response now. So the system basically bifurcates into two separate populations of phosphorylated and unphosphorylated substrate, where the phosphorylated population is much, much higher. And actually what happens in cells is that these phosphorylated and unphosphorylated substrates differentially control other parts of the pathway underneath them. So the response to a very small initial stimulus that modulates the activity of the kinase or the phosphatase will produce a very, very large response in the cell, which is again very hard to get by kind of using normal enzymes and concentrations of their substrates and things like this. And if I do the converse, which is to change the fraction 9 down to 0.9, so now there's slightly 10% less kinase than there is phosphatase, you get the exact opposite effect. And so this is a very, very simple model, as I showed you. It's, just go back here. This model essentially is just the set of rules are these 1, 2, 3, 4, 5, 6 rules here, with a set of rates and initial conditions, and you're able to model this pathway really nicely, or this motif in a pathway. And we've actually built much bigger models. We've used kappa for, we did a project with Pfizer building a virtual hepatocyte. There's something they were interested in, glucose metabolism. We built an insulin signaling model. We've built models of EGFR. So that model, the pathway I described with 10 to the 33 states, can actually be built with kappa from about 1600 rules, and you can actually run it and build it at full resolution. So it's quite remarkable. We also built models of dopamine signaling pathway, which was a sensitivity study for a company that were interested in neurotransmitters and drugs that modulate the effects of neurotransmitters. And as I said, Alex and myself right now are working with clients. We're actually working on a model of the immune system right now in a disease that it has a big autoimmune component. I can't really talk too much about it because I'm under the terms of a confidentiality agreement, but we are actually using kappa right now for real paying clients. So that's a little shout out to our little digital biology consulting company there. And then I had to finish with Derek Zoolander, of course. And that's it. Thank you.
Thank you.